Good afternoon and welcome HD research community. This webinar is dedicated to researchers, professionals, and the pharmacy industry. Thanks for joining us. I'm Professor Luciana Munhoz, a researcher from São Paulo University and the coordinator of the Scientific Committee of the Brazilian Union for Huntington Disease. I'm representing the Brazilian Huntington Disease Association today, or ABH as we call, which is sponsoring this webinar. Special thanks to Mr. Pedro Henrique Palma from ABH for organizing everything. This afternoon, we are presenting the research that won a prize at MENA Conference for Rare Diseases, named as Pharma Intelligence and Huntington's Disease, Unveiling New Treatment Horizons, developed by Professor Rodrigo Araldi and Professor João Rafael Dias Pinto. Professor Rodrigo Araldi, who is the speaker, holds a PhD on biotechnology from the University of São Paulo, with postdoctoral training in molecular biology at São Paulo Federal University, and he has been investigating neurodegenerative diseases as Huntington diseases for more than 10 years. He is the founder and the scientific director of the BioDecision Analytics. Professor João Dias Pinto holds a PhD in Sciences from São Paulo University with a postdoctoral training in Pharma Data Analytics at São Paulo Federal University. He worked at the Department of Earth, Planetary, and Space Sciences at the University of California, United States, and he is the professor in MBA program in Business Analytics and Big Data. He is the co-founder and the Artificial Intelligence Director of BioDecision Analytics. Professor Rodrigo Araldi will initiate the presentation, and you can send any questions you want in the chat. We will choose some of them for being answered in the final 10 minutes of the presentation. Professor Araldi, welcome.
Thank you, Luciana. Please, I'd like to only confirm that can you see my screen? Okay,
yes,
perfect.
Yes.
Thank you so much. Good afternoon for everyone. It's a pleasure to stay here. And before I start this presentation, I'd like to thank so much for the Brazilian Huntington Association, or Associação Brasil Huntington, specifically to Dr. Luciana Munhoz, who is scientific coordinator of this team. It's a pleasure to have this opportunity to share our data with all Huntington community, including scientists, pharmaceutical companies, patients, and everyone that is interested in this important field of research. Here, the main purpose of this webinar is to share with you the results that bring new hope for our patients, bring a complete study for the disease, discovering many potential drug targets and biomarkers for the disease, using pharma intelligence, a new novel technology that you can apply for the context of many other diseases. So, as you know, let me start the presentation. As you know, the Huntington disease is a neurodegenerative disorder that causes progressive neural loss, causing motor, neuropsychiatric, and cognitive dysfunctions. Although we know that the disease is very complex in nature, it's very important to remember that to date, we have no specific treatment approved that it can be postponed the natural history of the disorder. But this question is very important to be discussed. Since the first report of the disease that occurred in 1872 in Long Island, USA, and considering that the causative mutation that led to Huntington was only discovered about 120 years later. Today, we have more than 28,000 publications in literature describing different aspects of the disease. In terms of clinical trials, this number is very interesting. We have to date 256 clinical trials, most of them terminated or complete and part of them ongoing for Huntington disease. All these studies involving more than 177 subjects in different aspects of these clinical trials. If you think about clinical trials for drug development that comprise the most part of these studies, more than 12,000 patients are enrolled in these studies. It's a high number of subjects involved in these studies and a high amount of clinical trials ongoing. But the main question is why we don't have a specific treatment that can be changed the natural history of the disease. Analyzing these clinical trials, we can observe one important thing. The main criteria that are used that is used by these studies as primary or secondary outcome in these clinical trials is only focused on the Huntington scale. Although very important in clinic for analyze our patients, this scale is very subjective. And for this reason, it's very complicated to affirm the benefits promoted for one drug based only in this scale. For this reason, and you know that all scientific community is find for potential biomarkers that can help us to provide information about the benefits promoted for one drug in investigation. In a current published work from group of Dr. Sara Tabrizi, one of the most important researchers in the field of Huntington, we know that these observational scales are very subjective, and they prone to bias and inter-rater variability. For this reason, we are find for more sensitive, robust, and objective methods to track in Huntington disease progression. And this is very urgent for us in all science. But why there is no specific treatment? As you know, we have different technical aspects that can limit the potential to develop new drugs for Huntington. The first of one is the lack of proper animal models to preclinical studies. In the second point, we have no prognostic targets or biomarkers that can be used to provide the efficacy evidence of these investigational products in clinical trials, limiting these benefits as primary or secondary outcome in the use of Huntington scale. And third, and I think that the most important aspect that justify the lack of these specific treatments that can modify the natural history of the disorder is the lack of therapeutic targets or drug targets that can serve for us to develop new drugs in what we call drug design, and then can be pharmacologically modified to promote benefits for our patients. In this regard, we know that its biomarker is very important to monitor the Huntington disease progression. And in this sense, we know that the transcriptional dysregulation is a very important hallmark of Huntington disease, making the transcripts, the RNA, one important source of information that we can analyze to obtain a potential list of drug targets or even to identify these potential biomarkers. Specifically, if you are focused on RNAs that encode proteins, because as an important criteria for us to define one drug target or even a biomarker, it's important that this measurable molecule may be one protein. So we have also investigated these biomarkers in peripheral blood. Many studies try to identify its potential biomarkers along this data. However, it's very difficult to identify its potential biomarkers. There are several proteins, genes that are related to the disease, but in practice, none of them can be used because in other investigational studies or validation process, we cannot correlate, appropriate this biomarker with features of the disease. This is because all of them, of these studies that already published investigating these potential biomarkers are focused on microarray or DeepSage or even an RNA-seq analysis, but without no correlation with clinical pathological features of our patients. Based on this, we understand that the high-throughput sequencing technologies, particularly the RNA sequencing or RNA-seq, as in abbreviation, emerge as a useful technique to analyze the global gene expression to identify therapeutic and or prognostic targets that can serve for us to bring hope for our patients. In this regard, to date, several studies are already performed and available in literature that aim to assess this genetic information, this transcriptomic data of these patients in order to identify these potential targets. However, these targets identified by these studies cannot be appropriately used for us, not to drug development, either to make the use as a biomarker. And this occurs because reanalyzing these potential targets previously identified by literature, when you access the level of expression of these genes identified as targets, we observe that, yes, these markers are differentially expressed when you compare statistically with our control samples that are individuals without Huntington. However, the level of expression of these potential targets are less than 10. 10 is considered and recognized by the literature as a minimum expression limit to infer that this gene can be encoded in a protein. So most of these potential targets identified as a target gene for Huntington is not appropriated. Aiming to understand why this occurs, we reanalyzed the first study published that investigated transcriptional changes in Huntington patients. And reanalyzing this study, we create three possible hypotheses that can justify the results that lead to the comprehensive knowledge about why these potential targets previously identified cannot serve as drug target and biomarker. Our questions that we formulated based on the reanalysis of this study are: can the use of inappropriate control samples with no age matching with cases lead to target misidentification? And second, can the reduced number of AG gene positive individuals lead to target misidentification? And the third, can the lack of clinical pathological correlation negatively impact an accurate target gene identification? Aiming to answer the first question, we observe one important characteristic of this study. When you compare brain tissues, transcriptome profiles of Huntington individuals with controls, we observe that the age of these control individuals are statistically different than our Huntington patients. Basically, what these graphics mean is the control individuals are older than our Huntington patients. This is very interesting because when we reanalyze the samples and study this unmatched control from this analysis and compare the transcriptome profile of Huntington individuals with age-matched controls, we observed important results. Part of these genes that were previously identified target genes, when we split these patients that are older, these individuals, that are older than our patients, we observe no statistical difference between the level of expression. Justifying why, the reason because all of these genes previously identified as target genes are not used for us for clinical trials or even as a biomarker or even as a potential drug target. And this occurs because one very simple question. We know that Huntington disease naturally exacerbate or accelerate different aging-related molecular processes. This is one interesting reveal that describes all the systemic, cellular, and molecular alterations that aging promote. And interesting, we know today from different publications and studies already performed that Huntington disease naturally modify all of these parameters in an aging-like manner. For example, at systemic level, we know that the alterations, the systemic alterations promoted by the Huntington disease modify nutritional aspects of the patients, including involving the gut cells. More than this, at cellular level, we know that Huntington disease promotes cellular senescence as aging. The same occurs leading to stem cell exhaustion in the brain, specifically in the subventricular zone of the brain. and also induce the altered intracellular communication, modifying the communication between the cells. And this occurs mediated by extracellular vesicles and even by molecules that are directly produced and released into the intercellular space. At molecular level, we also know that the Huntington disease also contributes genomic instability, which is also verified in aging process, promoting a telomere shortening, even causing epigenetic alterations similar to those observed in aging. We have very molecular phenomena like as loss of proteases and even compromised autophagy observed in Huntington patients as observed in aging. And as very known for literature, the disease causes a mitochondrial dysfunction that is a hallmark and well-stated hallmark of aging. For this reason, we can demonstrate in this publication that was published about 1 month in Frontiers in Genetics that the use of control samples older than our individuals with Huntington disease can affect this interpretation and can lead to the misidentification of these potential target genes, justifying the results and the lack of these potential drug targets and the biomarkers that we are discussing along this presentation. However, although these important publications have been demonstrated how the use of these inappropriate control samples can affect our transcriptome analysis, two other questions may be to solve it. Can the reduced number of AG gene positive individuals lead to target identification? And can the lack of clinical pathological correlation negatively impact and create target gene identification? For answering this question, we collect samples from public database from the Sequence Read Archive, a public repository from the USA that have been collecting data from genetic studies, including from RNA-seq analysis. With this, we conduct the largest AG transcriptome study ever made, reanalyzing 353 samples, from which 146 samples were collected from AG-positive individuals. This is very important to remember that all previous study that we have in literature was focused on the analysis of only 5, 10, 7, or at least 18 samples. For this reason, we combine all the samples to increase the number of individuals. This is very important because, as you know, in terms of genetics, human population has important heterogeneity, and you need to understand these aspects in terms of the identification of these potential drug targets and even biomarkers. But we also understand that one important aspect that the technology available to date cannot be solved is the correlation between this genetic data with clinical pathological features. For this reason, we developed a novel innovative technology called BDASeq. BDASeq is a novel technology that combines bioinformatic tools, including the basic tools that are needed to perform one transcriptome analysis, but this technology has some difference to other techniques that are traditionally used. The first one is that we are not using here one only differential analysis technique. We combine 8 different techniques to analyze the differential expression. All of these 8 techniques is very well consolidated in the market and very well used, not only in science, but also by pharmaceutical companies. However, here we combine all these results using an innovative algorithm that it can be appropriate, select, and reduce the pros and cons of each one of these techniques. And other important aspects of this technique is that we are using artificial intelligence methodology. When I'm talking about artificial intelligence, I'm talking about statistical techniques very well consolidated for 2 decades that have been applied, appropriate, and combined with our genetic data can take together for this analysis important aspects of clinical pathological features of our patients, identify potential targets for different aspects of the disorder. Based on this, the first aspect that I have included in our technique is the propensity score matching, a statistical technique that appropriates select control samples according to features that we understand that are relevant for our study. As we demonstrate in our published paper, we verify that age of these control individuals can lead to the misidentification of these potential target genes. For this reason here, we select control samples appropriate according to the age of these patients. So, analyzing these more than 300 samples, and specifically the age distribution of our patients, our control individuals, we observed a statistical difference between the ages. This is exact science. We are using here post-mortem samples from control individuals. Based on this, we are analyzing the samples, and using the propensity match score, we can observe that this control group is not appropriate for our analysis. Demonstrating this, we conduct the transcriptome analysis, obtain the transcriptome profile of each subject involved in this study. And using a dimensional reduction analysis of this data, it's very clear to observe here in this first graphic, for example, that in blue you can see the samples of our patients with Huntington, and in gray the control samples. It's very possible to observe that there are many different transcriptional differences between this transcriptome profile between individuals with Huntington and control individuals. However, performing a clustering analysis, we detect that whereas our group of Huntington patients remain grouped in one cluster, the gray samples clustered in two different clusters. Analyzing the main difference between this cluster, we detect that the patients with Huntington that are comprised in the cluster 2, he referred in our study, are grouped in one only cluster, but our control individuals are grouped in two different clusters. And analyzing the main difference between this cluster, we can observe that the cluster 0 comprises the most of control individuals older than those that are comprised in cluster 1. Based on this and using the propensity match score that are involved in our technique, we excluded for our study these older control individuals that here are called unmatched controls. And so we compared the transcriptome profile of individuals with Huntington and controls, analyzing first samples from the brain, from a specific area of the brain, the prefrontal cortex, that, as you know, is directly affected by the disease. So we proved by statistical techniques that the age-matched control group and Huntington individuals, there is no statistical difference in terms of age, demonstrate that this group is now appropriate for our study. Next, we performed the differential expression analysis using our innovative algorithmic called recursive math combination that is implemented into the BDASeq. Using this technique and combined with AI algorithms, we identify a potential list of genes. One important difference of our techniques than other traditionally used in the market is that we can classify all the transcripts that can be encoded by the human genome according to different classes, such, for example, genes that are not expressed, that you call zero-count genes or low-count genes, that are those genes whose expression is less than 10, according to our first report that we identified that part of these genes previously identified in literature as potential target in summary comprised to low-count genes. Genes commonly expressed equally expressed or non-relevant genes, and these genes that we classify as upregulated or downregulated according to our algorithms that are the group of genes in which we can identify our potential target genes that can serve for drug design or as a biomarker. Based on this, our technique identifies more than 3,000 genes that are differentially expressed in the prefrontal cortex of individuals with AD. However, we have one question. How many of these genes are potential targets? It's very difficult among 30,000 genes identify who gene or how many genes are potential targets. For this reason, using artificial intelligence algorithms, we identify from these differentially expressed genes a total of 394 genes that can serve as a potential target. However, it's very important to validate these results. Here we are discussing about one artificial intelligence techniques that need be validated. For this reason, we analyze the expression level of these specific genes comparing the prefrontal cortex of individuals with Huntington whose use that not carry the mutation that lead to disease. And interestingly, here we can demonstrate in this heat map analysis that those genes that we identify that are upregulated and serve as potential targets are in fact upregulated in patients with Huntington in relation to our controls. By the contrast, those genes that we identify that are downregulated, we demonstrate that in fact are downregulated when compared to our control individuals. But we understand that only this evidence is not enough to prove that these genes are potential candidates to drug development or even to serve as a biomarker. For this reason, we perform another bioinformatics technique, in order to verify what these genes are affecting. What is the main pathways in these genes are participating, and how these biological pathways can be related to the signs and symptoms of the Huntington disease. Interesting, we observed that these 349 target genes identified by our artificial intelligence and validated are closely related to processes such as neuroinflammation, transport across the brain-blood barrier, metal-ion response, cholesterol metabolism, axonal guidance, neurotransmission, leading to behavior or neuropsychiatric symptoms. We know that all of these molecular pathways are closely related to the pathophysiology of Huntington disease, demonstrating and confirm that the artificial intelligence used in our technology was able to accurately identify potential target genes that can be pharmacologically modified. We also demonstrated that these specific genes that we are demonstrating and can serve as drug target are involved with aging. This is expected because, as you know, the disease, Huntington disease, exacerbates the aging process. So it's expected that the target genes can be related to the brain aging. And specifically, this is what we observed in our results. We detect that the genes that are differentially expressed and serve as target identified by our technique can accelerate the aging process, not only in the brain, but at a systematic levels. We have several publications in the last decade demonstrating the systemic effects of Huntington disease. Most part of these effects, systemic effects, are closely related to the acceleration process of this aging. And so here we are demonstrating that the agents that govern this process is specifically the set of target genes identified by our study, in which we can observe that they control the aging of brain, bone marrow, muscle, adipose tissue, immunological systems. This demonstrates for us that we have potential biomarkers not only to be employed for treatment for Huntington, but for other diseases, and even to postpone the natural process of brain aging that it can be a prophylactic potential therapeutic in the future to avoid some diseases like dementia that you know is a very important global burden today in terms of health. Interestingly, we also observe that this target gene is identified by our algorithms. It's also differentially expressed in different brain areas beside the prefrontal cortex. But interesting, these specific brain areas that we are demonstrating in this heat map, for example, such as cingulate cortex, striatum, neuron, glial cells, astrocyte, caudate nucleus, thalamus, motor cortex, substantia nigra, cerebellum, pontine, it's also related with the evolution of Huntington disease. So if you can modify the expression of these genes, it's possible that we can postpone the evolution of the natural history of Huntington disease. Moreover, you know that these brain areas are not involved in Huntington. These brain areas are also affected by other neurodegenerative disorders, such, for example, frontotemporal ataxia, Alzheimer, Parkinson, amyotrophic lateral sclerosis, and other neurodegenerative disorders, making possible that the same targets can be useful to be the treatment of other disease. And this is expected. Science is, you know, Huntington disease is considered and recognized an important model to study different types of neurodegenerative disorders. Confirming this result, we also investigate the expression level of these same targets that we identified for Huntington disease for other neurodegenerative disorders. And we confirm using data set from this disease that these genes are differentially expressed in Alzheimer, Parkinson, Friedreich ataxia, dementia, sclerosis, autism, bipolar disorder, schizophrenia, demonstrating that it's possible to employ the same results for the treatment of other diseases. However, the main focus of our study is based on to describe the potential target genes for Huntington. And for this, the main difference aspect of our study compared to all of them already published is that we connect the clinical pathological features of our patients to identify biomarkers or therapeutic targets that can be related to several important aspects of the disease using artificial intelligence algorithms. For this, first of all, we analyze the clinical pathological features to understand what of these clinical pathological features are relevant for this type of analysis. For this, we use the BDASeq, another analytical technique developed by our company, to identify relevant clinical pathological features of interest that can be connected to genetic or transcriptomic data to identify these potential drug targets and biomarkers. Using the BDASeq, we identified one important clinical pathological feature is the CAG repeat length. This is very well stated in literature. We demonstrate that the longer the CAG repeat, the early the onset age and that of age of our patients. And therefore, as expected, the less the onset, the less that age of our patients. Demonstrate that CAG repeat length is an important clinical pathological feature. For this reason, using artificial intelligence, we describe for the first time a set of genes that are downregulated and upregulated and can be closely related to the CAG repetitions. So today, using the expression level of these protein coding genes, we can determine the evolution of our patients and correlate this natural history or the evolution of our patients in terms of the CAG repetitions of these individuals. Exploring these clinical pathological features using the BDExplore, we also demonstrate that there is no gender difference for the onset age of these individuals. But considering the onset grade, that is a scale to analyze the neurodegeneration, we observe that the higher the neurodegeneration, the early the onset and the dead age of our patients. But larger the CAG repeat, emphasizing that the CAG repeat length is very important and are directly related to several aspects of the disorder, including neurodegeneration. Based on this, we also identify using artificial intelligence novel potential drug targets and biomarkers that can be related to the level of neurodegeneration of the brain. So today we have and provide a complete list of both down and upregulated genes, which expression levels can be related to the level of this neurodegeneration and can serve for us to understand the evolution of our patients in terms of natural history. Use the same approach, we also identify potential target genes that are related to the onset age of these patients, identified both down and upregulated genes that it can be justify why some patients develop the disease early and other later. We know that the CAG repetition is related to this time of onset age. However, we don't know what are the molecules or the genes that are differentially expressed as a causative or a consequence of this CAG repeat length. Here, we are providing for the first time the potential candidate list of genes that are related to the onset age. And importantly, based on all these informations, we identified a list of down and upregulated genes that can be easily pharmacologically modified by both drugs approved for another disease and already available in the market, and can be also explored for the development of new drugs, including biological drugs. And if you can modify the expression level of these target genes, we can increase the lifespan of our patients, supposedly postpone the neurodegeneration. In this regard, we also analyze it in terms of drug banks already approved in the market, the potential drugs that we can use or reposition from the treatment of Huntington disease. We know that we have a lack of drugs in the markets. Today, only tetrabenazine approved by FDA and other international regulatory agencies is used with some benefits approved by the FDA, and many other compounds and drugs are in investigation. In terms of these investigations, as you know, all of these results is focused on the Huntington scale as a clinical scale and subjective study. But here we are demonstrating that other potential drugs could be used for the treatment of Huntington. One example of this drug is valproic acid. Interesting, there are many studies in literature demonstrating the potential of valproic acid for the treatment of Huntington for more than 2 decades. However, there is no clinical trial for the analysis of the potential benefits of this drug for the treatment of our patients. The main purpose of this webinar is not directly the specific type of drug or lead one investigation of one of these drug targets. The main purpose is to provide a list of target genes that can be modulated by drugs or biological treatments, such, for example, stem cells, that can be employed in the future for the treatment of our patients, but also to provide biomarkers, most useful biomarkers than the Huntington scale, that can be used in our clinical trials to provide the evidence of the benefits promoted by these investigational products. Based on this, it's important to remember that it was only possible to identify these potential targets because the use of our technology. This point is not to emphasize the importance of our technique. This is a not marketing presentation, but this is to justify why other studies cannot identify these potential targets. The main reason of this is the lack of combination of different technologies to analyze the bioinformatic process related to transcriptome analysis, and also the lack of clinical pathological features that are involved in these studies. With this, we identified about more than 800 genes that was not previously identified as differentially expressed. And we identified, using our technique, about 1,300 genes that serve as a potential target for drug design or even for biomarker, since these genes are protein coding genes. This occurs because our technology is the only technology able to simultaneously control the error type 1 and type 2. In other words, to control the false positive and false negative results, that is the pros and cons that you can observe in all of these bioinformatic tools. When you combine different techniques and use appropriate intelligent algorithms that we implement in our BDASeq technology, we can control the false positive negative errors simultaneously, leading to a reclassification of these genes that lead to identification of these potential biomarkers that were described in this result and was validated in our study. But when you're talking about a biomarker, we also understand that this analysis and all the results that I commented before are focused on prefrontal cortex. And to analyze a type of sample like this, we need to collect a brain tissue. For this reason, this is not appropriate biosample for our purpose in terms of clinical trial investigation. We need to have one biomarker that can be easily collected. In these terms, the blood is the main and most interesting tissue to be analyzed the expression of one biomarker. For this reason, using the same technology, the same approach that used to analyze the prefrontal cortex, we also investigated the presence of these potential targets in peripheral blood of our patients, analyze the transcriptome of peripheral blood. And interesting, we identify from thousands of genes, one gene that is differentially expressed in the blood of patients with Huntington. Interesting, this same gene was identified as a potential target in the brain. If you consider the concept of one drug target and a biomarker, it's important that this biomarker need to be a molecule, typically a protein, and this protein may be related to the disease. And maybe relate disease affect the main organ or tissue that are affected by the specific disease investigated, in our case, the brain, and can be detected in the blood. So here we are providing one biomarker for the first time that can be identified. These biomarkers are closely related to the onset age, to the lifespan of our patients, to the neurodegeneration, and play a key role in processes involving oxidative stress and neuroinflammation. This biomarker identified in blood of our patients, we observe that it is downregulated in the brain, and this downregulation lead to the neurodegeneration, and this downregulation occur in a CAG repeat length dependent manner. But interesting, we observe that expression level of this biomarker is increased in peripheral blood. Interesting, this increase of this protein in peripheral blood was not seen in literature in 1972 for the first time. However, due to the lack of another corroborative studies later of this publication, mainly of those focus on RNA-seq analysis or even in microRNA analysis, these biomarkers was lost in literature. Among the more than 28,000 publications about Huntington disease. but we have proof that we can measure this protein in order to use this biomarker as a potential biomarker for our clinical trials, to investigate and help the pharmaceutical industry to provide the benefits promoted by their investigational products for Huntington disease, including one biomarker that is not subjective, as, for example, the Huntington scale today mostly used in all of studies due to the lack of this biomarker. So, to finish the presentation, I would like to conclude that our technique was able to identify potential therapeutic targets and biomarkers and can be used not only for Huntington, but for all types of disease, particularly to rare disease in which we have a limited number of samples to be analyzed. In terms of Huntington disease, our technique was able to identify about 400 targets, validated targets, that can be served as a drug target or a biomarker, helping us to develop a new treatment, helping us to investigate the potential of this treatment in our patients, providing evidence of its benefits in clinical trials. So all these results demonstrate that these targets, it's very important, bringing hope for our patients, hope for Huntington, and even for other diseases, for example, Parkinson, Alzheimer, and other diseases. It's very possible that we can obtain single drugs in the future that can treat all of these diseases together. So from these 394 targets identified by our technology, we can provide a list of genes that can serve as targets related to brain generation, lifespan, that can be detected in the blood-serving biomarker, being related to onset age and CAG repeat length. To conclude, I would like to emphasize that all of these results have been not seen in Brazil, in Latin America, and world, in different magazines, journals, demonstrated the potential not only of the technology, but specifically the new horizons that we are bringing for our patients with Huntington. It's important to remember, and for us a pleasure to not see that both our technology and the results obtained with our technology was awarded in the MENA Congress for Rare Disease that occurred along this year in Abu Dhabi. This is the most important and the largest event dedicated to rare disease in the world. To finish, I would like to thank you, my team, especially Professor Dr. João Rafael Dias Pinto, who stayed with us in this presentation, and that together with Benedito, who is our statistician, was responsible for the development of all these algorithms that bring to us this potential BDASeq technology and was responsible to validation of this data. I would like to thank you so much for international and national institutions that support our studies, including International Huntington Association, Associação Brasil Huntington, Federação Brasileira de Alzheimer or Alzheimer Brazilian Association, the university in which I am located, Paulista School of Medicine from Federal University of São Paulo, FAPESP, that was responsible for the financial support to this analysis, Google Cloud for startups, and Información that represent Google in Latin America. I would also like to manifest my acknowledgement for some persons that are very important in this study and for all the scientific community in Huntington disease. Dr. Luciana Munhoz, that is coordinator of this team of scientists in Huntington. Luciana, thank you so much for all your efforts to make this webinar possible. Professor Dr. Irina Kerks, who is a geneticist specialist in Huntington disease with 3 decades only dedicated in Huntington disease. Professor Irina Kerks is an inventor of one important technology based on stem cell for the treatment of Huntington disease that is now beginning in phase 3 clinical trial. Thank you so much, Irina, for all your support, for your acknowledgement, and all about our discussions along this time that lead to conclude all these results. Dr. Joyce Macedo, neurologist, specialist in Huntington, and PI of studies involving clinical trials involving product for Huntington, and Katia Favalli, who is coordinator of PESP, and by being a pharmaceutical, is participating of all this study and have the opportunity to discuss about of our technology. Thanks so much, and it will be a pleasure to answer any question.
Thank you, Professor Rodrigo Araldi. We will start doing some questions that were already being done before, were sent for me by Brazilians, and I translate to English. And those, I think they are very important to be recorded in this version of the webinar, okay? First question, what will be the next steps?
Thank you for the question, Luciana. As we're talking here, the main purpose of our company for this study is to identify these potential targets. This is the main focus of our study and our company. We are not a pharmaceutical company, so we cannot lead this data and this information we don't have the support of the pharmaceutical industry. So the next step is to lead all these results, all this information from the pharmaceutical companies. We understand that using all this data, it's very possible to reposition some drugs from patients with Huntington, making an offer support for some signs and symptoms. Maybe it's possible to develop new drugs, new technologies that it can be used for the treatment and can be modified in natural history of Huntington.
Thank you for the answer. The second question is, how can this study contribute to other rare diseases?
Perfect. We understand that Huntington is a rare disease, but in terms of amount of patients and samples that you can study, we have more samples than other diseases that are very most rare in terms of number of patients. Here, we demonstrate one technology that it can obtain available information and possible drug targets and biomarkers for rare disease, make it possible to explore many other rare diseases around the world. And we understand the main problem when you're talking about orphan drugs. This rare disease have one very difficult problem in terms of drugs. We don't have the interest, maybe for pharmaceutical companies, even the reduced number of individuals affected by the disease. And even we have some limitation. It's very difficult to identify a target gene, a target molecule to develop one new drug. So these results demonstrate the potential to develop novel drugs from these orphan diseases. So the applications of BDASeq, BDExplore is not only limited to Huntington, but it can be used for all other diseases, including rare disease.
Thank you for the answer. The third question is, why blood marker is so important and why it is so difficult to find a blood marker?
Okay. To find a biomarker is a very difficult process. For example, in oncology field, only 1% of all biomarkers already identified to date can be used for drug development or can be used for diagnosis or even differential diagnosis in cases of cancer. This occurs because maybe you can identify a protein, a molecule that we can recognize as potential biomarker, but reanalyzing and at the moment that we increase the number of patients due with the genetic heterogeneity observed in human population, we can observe that control individuals or persons that don't have this specific disease have the same expression level of this biomarker as someone that you previously identified that have this gene overexpressed or downregulated gene. So in terms, when you're talking about a technology like this, we can identify these target genes. We can avoid this misidentification of these potential target genes. And when you have one biomarker or a target gene, I think that is the most simple word here, we can develop new treatments and you can understand if this potential new treatment is effective for the treatment of the disease. This is one of the most difficult points in our clinical trials. How we can confirm that the drugs in clinical trials ongoing are effective if the only matter to analyze this phenomenon is based on a subjective scale. I'm not talking that this scale is not enough. This scale is very important in clinic for all physicians, but we need for more information for these clinical trials. This will help pharmaceutical companies to provide evidence of these benefits to regulatory agencies, facilitating the registration process.
Okay, I will meet a question from the Vice President of the Brazilian Huntington Disease Union House, Mr. Marcos Ferreira. Which drugs you found to be useful in HD treatment, sorry.
Yes, thank you for this question. We identified using a public database for drugs, a set of potential drugs that can be explored for the treatment of Huntington. Obviously, here I'm not emphasizing that this drug are effective. We need to perform some clinical trials to confirm these results as make part of the regulatory process to approve any kind of drug. Among these drugs, I emphasize that valproic acid is one of the most potential drugs to be applied. Is potential effect treatment by affecting multiple genes simultaneously and can be promote different effects, affect many types of biological pathways that are deregulated. It's possible that valproic acid, that is a very common drug and used for a long data, and important to remember that already tested in studies, but only in scientific literature, and demonstrates some benefits for patients. But if used in correct posology and dose, we can promote most and interesting benefits for our patients. Moreover, we understand that the genes that we identify here in our study that are downregulated can be easily modified by biological products. For example, with stem cells. Stem cells is a great opportunity for trait patients. We understand that stem cells can contribute from bringing benefits for our patients because these cells express many of these genes that are downregulated in our patients. Can be promoting one compensatory effect from these genes that the disease reduce in terms of expression. So we have two different potential to use drugs, the biological products, or even both simultaneously in the future. In terms of drug reposition, we can defend the use of valproic acid. Remember, we need clinical trials to demonstrate this effect. We need to understand the posology, the drug concentration. And also, we have the opportunity using these biomarkers to develop new drugs. It's important to remember that the drugs that we're present in here in this webinar, it's only a few and reduced number of potential drugs. There are many drug bases in pharmaceutical companies that can be easily explored, understand what is a specific target that it can lead to identification of most powerful drugs for the treatment of Huntington.
Okay, the last question. Why the Unified Huntington's Disease Rating Scale is not suitable for outcome variable for clinical trials?
Perfect, because the scale is subjective. The scale is a report of one physician. In terms of regulatory agencies, it's also it's not obligatory. There is the reason because we are using the Huntington scale in all of these studies, and we need to remember that the use of this scale here, I'm not saying that is a mistake. we have understand that to date, we have no biomarker in literature. We don't have any biomarker described in science or previously validated. For this reason, it's very difficult to use a biomarker if you don't have a biomarker. But what this company is using is a scale, but this scale is a clinical scale. So this is a report's impression of one physician, that you know that if these teams of physicians or the team of persons that are involved in the application of the scale in this patient are not appropriate trainee in this scale, or even if there is some points of these scales that it can be changed along the evaluation. The points of this scale is very subjective. The difference between one and other point is very close, and for these reasons, very difficult to determine if the patient today is point 1 or point 2, for example. And this subjective can lead to some misinterpretation of this data. More often, the scale analyzes different aspects of the disease. And one part that is very important to be discussed here is that when we analyze clinical trials, we can observe that different companies are using different points of these scales. For example, there are companies using the motor score scale, other companies using the cognitive scale, others the chorea score, others are using the total function capacity. But there is no unified parameter to analyze the point of these drugs. We also understand that it's very difficult because there are drugs that are being evaluated to analyze with the objective, with the goal to promote one cognitive benefit. So we understand the primary goal, the primary outcome to be analyzed in these patients, obviously, is the cognitive aspects, another, the motor aspects. But we have many drugs to be ongoing in analysis now for motor function, bring to avoid or to reduce the chorea of our patients. But these clinical trials are investigating different aspects of the scales. We don't have one unified parameter. For example, total motor capacity, total function score, chorea score. What is the aspect that I need to analyze? This makes it very difficult to compare all of these clinical trials, all of these drugs that have been analyzed. This is not to prove that have a mistake. It's a problem. It's a problem for pharmaceutical company that don't have these types of biomarkers. But now we have a potential biomarker. We have the possibility to aggregate information in our clinical trial using a biomarker that can be analyzed in peripheral blood of our patients using a biochemical assay, for example, ELISA, to detect the level of one protein. And with this, to provide a benefit potential of this drug.
Okay, thank you. We have one more question here. How do the pharmaceuticals companies could use the artificial intelligence you developed for drug discovery and outcome testing?
Perfect. Dr. João would like to answer this question, a specialist in artificial intelligence.
Yes, maybe I can help you. So the technology we developed can help to find targets by combining data from studies, from transcriptomic analysis, and aggregating patient level data to find the most important features connecting to outcome in a variable of interest. So the pharmaceutical companies can benefit, in fact, with the technology because we deal today with a large amount of data with many variables. So the traditional statistics approach that is based on p-value sometimes are not enough to identify patterns, the underlying patterns, the connection of the main variables and the variables of interest and the main attributes. in this study. So the artificial intelligence can be a very important tool today for analyze large amount of data with a lot of variables and can produce very important results.
Thank you. And it's important to remember that artificial intelligence and all these techniques is not a feature, it's a present. All these techniques is now used and have been used by different pharmaceutical global in Brazil to identify potential targets, to identify clinical pathological features to be analyzed in clinical trials. This is a present. And artificial intelligence, I understand that is a very interesting aspect of the innovative science, but it's the presence. Is what we are using in all of these studies. It's not a new technique, and what we're applying here is exactly we need to apply to discovery with precision to accuracy, novel target genes.
Okay. Do you have anything to complement or? Okay. Again, thanks for ABH, the Brazilian Huntington Disease Association, for sponsoring this webinar. The record of this webinar will be available at the YouTube channel of ABH as well. Thanks, Professor Rodrigo and Professor João. We'd like to have more time for talking about this. It's a really amazing subject. Well, we are going to finish the conference, and thanks for everybody that's joined us today. See you. Bye-bye.
Thank you very much.
Thank you so much.